Hi, everyone. Thank you for joining us for our May Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Hereditary Disease Foundation. I want to start by thanking our sponsors for this webinar, Neurocrine Biosciences, Cleveland Clinic Lou Ruvo Center at Brain Health, and Unicure. Also, thank you to everyone who supports the HDF and to all of you for tuning in. Thank you all for being here. I'm excited about today's webinar because it falls during HD Awareness Month, and we have two absolutely fantastic speakers who are joining us from opposing time zones in California and in London. They have a very fitting talk for this month. They'll be sharing a fireside style chat about how drugs get made and what's happening now in HD therapeutics. We're joined today by Dr. Frank Bennett and Dr. Ed Wild. Dr. Bennett is one of the founding members of Ionis Pharmaceuticals, leading much of their pharmaceutical antisense drug discovery and antisense technology research. He's been involved in the development of antisense oligonucleotides as therapeutic agents, including research on the application of oligonucleotides for inflammatory, neurodegenerative diseases and cancer, oligonucleotide delivery, pharmacokinetics, and medicinal chemistry. Frank collaborated with Adrian Krainer of Cold Spring Harbor Laboratory to develop Spinraza, the first effective therapy for spinal muscular atrophy, a progressive neurodegenerative disease and the #1 genetic cause of death for infants. Dr. Wild is a professor of neurology at the University College London, a consultant neurologist at the National Hospital for Neurology and Neurosurgery in London's Queen Square, and associate director of UCL Huntington's Disease Center. He leads a team of researchers aiming to accelerate the development of new therapies and make a real difference for people impacted by Huntington's disease. He gave the first injection of tominersen in 2015 and has been involved in the design and running of numerous HD clinical trials. He co-founded HD Buzz in 2010. Thank you both for joining us today. Before we start, I want to remind our audience that you can ask questions in the Q&A box throughout the talk. So make sure you're using the Q&A box and not the chat box. We will have time at the end for a Q&A with the speakers, so send your questions in as you think of them, and we'll get to as many of those as we can at the end. And also, this will be recorded and available on our website and YouTube channel in about a week or two. So if you want to rewatch it again, or if you want to share it with someone, it will be available. Thank you all so much for being here, and I will pass it over to Ed and Frank.
Hey,
thank you.
Thank you, Sarah. And welcome to London on my corner of your screen. So we, as you can see, are having a literal fireside chat. I should introduce also an observer. This is my dog, Riley, who sometimes cycles me to the office. So we're just going to kind of figure out where we've come from, I think, in terms of new drugs and clinical trials and the journey that we've been on that's brought us to 2024 and what we've learned and where we think we're going next and perhaps how we can make the future brighter than the recent past in terms of therapeutics. Frank has been doing this somewhat longer than I have. So it'll seem like I'm interviewing Frank to begin with, and then the tables might turn once we start talking about the actual conduct of trials and injections and stuff. So what I want to do, Frank, is ask you, well, so I started working in HD in 2005, and I have a very vivid memory on the London Underground on my first day to work for my friend and mentor, Sarah Tabrizi. She had given me a big red booklet of abstracts from the Hereditary Disease Foundation meeting in Boston, which had just happened a couple of months earlier. And I remember looking at a couple of abstracts which talked about something called gene silencing at that time and thinking, first of all, I thought that sounded amazing. And then second, I thought that I was really worried that we would have a cure for Huntington's disease before I would be able to finish my 3-year PhD. So one of those things was correct, which is that Huntington lowering and gene silencing are amazing concepts. So let me ask you, what is gene silencing, or how would you describe the kinds of drugs that you have been involved in developing for HD?
Yeah, so I've been working on developing what we call antisense oligonucleotides, which is one of the ways you can silence a gene. For this month is my 35th anniversary for doing it. And really what we're talking about is everybody recognizes that the gene is where we store our genetic information. That's transcribed into an intermediate molecule called RNA, and then the RNA is translated to a protein. And so you have this flow of genetic information from DNA to RNA to protein. And a very bright scientist in the 70s, late 70s, came up with the idea of using short synthetic oligonucleotides. So they're short snippets of our genetic information that are designed to bind to the RNA. And if it binds to the RNA, it prevents the protein from ever being made. So you're short-circuiting that genetic flow of information. So you make DNA makes RNA, then the RNA is degraded and no longer makes the protein. And so what we're developed is a technology that really utilizes the exquisite selectivity that occurs, Watson-Crick base pairing, or how DNA binds to DNA or RNA binds to DNA. And we're using that same exquisite mechanism for designing our drugs. And so what we're essentially doing is using short synthetic oligonucleotides that bind to RNA and can selectively degrade only the Huntington RNA or whatever the particular target is that we have designed our drugs to inhibit. And so it sounds simple. It's taken us 35 years to get where we are today, and there's still probably another opportunity to further improve on the technology over the next few years.
So the sort of underlying biology, I guess, was described in the 70s, but what was the sort of thinking that made people figure out that these could be drugs rather than just a sort of interesting thing?
Yeah, so I mean, it's really important to kind of put yourself back in 1978 when the first papers were published.
The year I was born.
Yeah. So, at the time, making an oligonucleotide took 3 postdoc years. So right. It was not simple to make those. And so there needed to be some technology that evolved. And in the 80s, 2 people, Leroy Hood and Marv Carruthers developed the chemistry that made it fairly simple to make oligonucleotides. And Leroy Hood developed what's called, at the time, the gene machine, where you can synthesize the oligonucleotides. And so that transformed our field and really enabled it. And then there were some chemical modifications. So wild-type DNA or natural RNA gets degraded within a few minutes of injecting it into the body. And so we had to use some chemical modifications to stabilize the oligonucleotides so that they would stay around long enough to do what we want them to do. And so it was really those 3 things that came together that by the late 80s, when we started Ionis, or ISIS at the time, different ISIS, but
we can come back to that if we have time.
Yeah. But it was really those 3 things that gave us the ability to start a company and really think about developing these as therapeutics rather than as research tools.
Right. So your formative years in the late 80s, early 90s then were sort of work towards essentially weaponizing DNA to become a drug.
Exactly, and learning where it worked and where it didn't work. So we had a lot of ups and downs in that period of time where we were really trying to figure out where the technology would work well and where it didn't work well. And it wasn't really until you started entering the field that we realized that the CNS is an area that the technology worked well. So that took a lot of time to get there, but once we got there, we were able to exploit it and started developing antisense technology for a variety of neurological diseases.
Yeah, so roughly 5 years after you started working on ASOs was the year that publication of the HD gene occurred in 1993. Was that a big moment for you at the time, or did it sort of pass you by as being sort of vaguely interesting and we can come back to it later when we're a bit further ahead?
It's a bit of both. So I had known about Huntington's disease. Many people don't know this, but I used to practice as a hospital pharmacist. And so I you would see Huntington's disease in the admission diagnosis for patients. So I knew about the disease. I didn't really know much about the disease, but I knew it existed. And when the paper was published in 93, I found it extremely intriguing, and it looked like that should be something that we should be addressable with our technology. It really took another 10 years for us to further optimize the technology for us to really seriously consider going after Huntington's disease. So there was still a little bit of an evolution that needed to take place. But I still remember the paper and reading that issue of Cell that described the gene. So it really was a landmark for the field.
But as you got towards having molecules that could be contemplated as drug candidates for humans, you must have, because HD was sort of pretty famous among these single gene brain diseases. That must have put it at, so I guess the question is, what is it about Huntington's disease that makes it a good target for these kinds of approaches?
I've been in the pharmaceutical business now for 40 years, so I'm aging myself a bit, but it really comes down to what is the molecular hypothesis? And so for Huntington's disease, it was very attractive to think about just lowering the mutant protein or prevent it from ever being made. Secondly is, do we have biomarkers to measure the drug effect? And Ed, you were heavily involved in developing the biomarkers that would allow us to demonstrate that the drug did what it was designed to do. And then third is also, in your realm, sort of the clinical endpoints. How do you measure whether the drug is producing an effect in patients? Yeah. And so, again, in 93, when this paper was published, a lot of this was unknown. And we had more work to do as a field, but by the time we started working on Huntington, I think it was roughly about the 2004, 2005 timeframe. A lot of that was coming together to enable us to go forward. And Ed, for your the work that you did on developing a biomarker was really transformative, and it gave us the confidence to go further. I mean, I'll let you talk about that. You should describe why you started working on that and the progress.
Oh, yeah. Well, okay. So kind of essentially everything that my whole career was basically founded by work that Sarah Tabrizi was doing for the 5 or 10 years before I started working with her. And she, as a clinician scientist, had been very focused on the idea that changes in improvements in drugs were likely to bring a new generation of drugs to HD, but if they worked, it would be really difficult to prove that they were working. And the example I always give is of someone who has an HD mutation or expansion, who's 40 years of age. If you give them a drug that works perfectly, 5 years later, they still look the same. But looking at them from the outside, or even doing sensitive clinical examinations like neurologists do, we have no way of knowing whether that's because the drugs worked or whether that person was going to still be fine anyway at 45. So biomarkers are basically anything that lets us look inside the body and figure out on a biological level what's happening with the disease, or even upstream of that, whether the drug is essentially hitting its target and engaging with its target. And I remember very clearly, so my initial work was on brain imaging with MRI scans, measuring the shrinkage of the brain, which we thought then and still think now could be a really useful way of showing that we've intervened in a meaningful way if we can slow the shrinkage of the brain. But during that process, we ended up collecting blood samples and then later on samples of spinal fluid or CSF, because there'd been a very memorable occasion where a man called Graham Bilby, who worked at Novartis Pharmaceuticals at the time, came to address a meeting of the European HD Network Biomarker Working Group in 2007 or 8. And he said, he gave his talk about how drug development works from the perspective of a big pharmaceutical industry. But then he said, just a quick question for you. What's the concentration of the mutant Huntington protein in spinal fluid? That's the cause of the disease. We know that because of the gene. How much of there is it in CSF, and how will you know if you've successfully lowered the production of the protein. And there was a bit of an embarrassed pause, because actually we had to then confess to this big important drug man that we couldn't measure it. Not only that, but we hadn't really systematically collected spinal fluid in order to begin the process of developing measurements. But of course, as soon as he said it, it became obvious that that needed to be a top priority, because we were also aware that people like you, Frank, were developing drugs to lower the protein. And you know, running a trial without knowing whether the drug is hitting its target over a time span of a few months ends up being a really challenging thing, because you have to in theory, you would have to go in blindly and run a trial for 5 or 10 years just to follow the clinical endpoints, and that's something that no drug company really has the money or appetite for. So and again, these things happen because of the effort that people put in, and then the stuff that just sort of lands on you from outside. And in our case, the two things that really fertilized well, let me say three things that fertilized the development of the first Huntington biomarker in the nervous system was, number one, the technological advancement in the field of what are called assay platforms, which is essentially measurement machines, which will you put a sample in, and it science happens, and a number appears on the screen that is the concentration of the protein. But the established methods using things like ELISA technology, antibody technology, those were not sensitive enough. And we knew that the concentration of mutant Huntington, which is released from tiny brain cells at a very slow rate, we knew that that concentration was going to be super, super, super low. So, but when these new ultra-sensitive single molecule counting machines came along, it was clear that the time was right to have another go. The second thing that occurred was that CHDI Foundation, and in particular Doug McDonald, really kind of put together a consortium of clinician scientists like me to collect the samples and sort of give a sort of clinical interpretation of what we might find, plus assay development people. And it was someone else from Novartis called Andreas Weiss, who spent 3 years developing a new antibody that would stick to the very start end of the Huntington protein. We already had another antibody called MW1, which stands for Milton Wexler 1, for the HDF fans in the audience, which is everyone. And that antibody was named after Milton. It turned out that those two antibodies, when you stuck them to the Huntington protein, gave a really strong and powerful readout when you fed that into the new assay platform. And there was I do have one or two visual aids with no slides, but I want to show you an email attachment that I have framed on my office wall. This is the email that I sent to the CHDI and Novartis collaborators when they sent back I had collected some CSF samples and we used the assay, and basically what you could see was that the assay was producing a really strong signal for mutant Huntington in the people with the mutation, and it was also related to what we call disease burden score. So the further on you were in the disease, the more Huntington was in the CSF. And this is a meme from about 15 years ago. It was very popular at the time. Suffice it to say that we were extremely pleased when we analyzed the results. That all happened and was finally published in 2015, which turned out to be the year when the drug that you had made, which at that time was called HTTRX, was ready to go into people. So, but there was the missing piece of the jigsaw now is that we knew that if you eat DNA, your stomach will dissolve it and it won't enter the bloodstream. If you inject it into the blood, most of it gets taken up by the liver. So how then did we go about figuring out how to get ASO drugs into the brain?
Yeah, so that was, we had recognized what you had just described, many, many years ago. And, I'm sometimes simple in how I think about problems. And so, there's been a lot of work, and there continues to be a lot of work for developing really sophisticated delivery technologies to get drugs into the brain. But given how the disease that we're dealing with and the needs for patients, I recognize the patients couldn't wait for the science to evolve to make a more convenient delivery system. So being a simplest, I said, let's just introduce it into the cerebral spinal fluid that surrounds the brain. And we started off doing that for a program for ALS that is now approved called tofersen. But that was our first model system that we were working on. And once we demonstrated that, surprisingly, you introduce the drug into the fluid that surrounds the brain, it very quickly distributes throughout the neural axis, and you start seeing drug in brain tissue. We reasoned that a lumbar puncture is medically tractable. So it's it's a little scary in the beginning, and I'll let you describe your experience with it. But it's it was something that clinicians were comfortable doing, and we opted to use that method as a delivery system. Basically, putting a needle into the spinal fluid as a way of delivering it, and it worked. And so we have continued to push forward with that.
It's funny you say clinicians were comfortable with it, Frank. They really weren't.
Yeah.
I would say that some clinicians were comfortable with the idea, but terrified at the idea that we would have to do it. Because honestly, unless you're really a hematologist, neurologist, dealing with things like lymphoma, or one or two other very specialized bits of neurology, these intrathecal injections, as we call them, into the spine, were not really something that an everyday neurologist did. And certainly if you enter Huntington's disease as a clinical specialty, you expect to be doing what you can to help people, but mostly not being able to. And most of it was fairly kind of low tech. We had some pills that would help with symptoms, and various forms of multidisciplinary therapy. The idea of injecting designer drugs into the spine to reach the brain was very alien, certainly to me. And I remember having to put on scrubs for the first time, surgical scrubs, and go to the cancer center in our sister hospital to get trained in how to do this stuff. And then so we were basically laying the railway track from the front of the train. It's another visual aid here, because the 5th of September 2015, which was 9 years and 11 months after I started in HD. This is me giving the first injection of the drug into the spine of a patient that I had already known for 10 years by that time. And the gold medal was given to me by your friend Anne Smith at Ionis, secretly at that time, because she was able to reveal that I had given the first dose of active drug. Terrifying, though, honestly terrifying. And obviously, the Ionis had done its due diligence in terms of proving that these drugs were as safe as they could be, but giving that first dose, or giving the first dose to any of our patients, was a really really stressful experience. But what I can tell you then is that the trial, once it was underway, quickly turned into a very sort of routine thing. 46 patients were enrolled at 4 sites worldwide. Was it 4 or 6? I think it was 6 sites worldwide.
Yep.
Say again?
I think it was 6.
Yeah. And, that first trial for each participant was very short, just 4 doses a month apart, and then a follow-up period. But it took 2 years to go from start to finish, because we were very cautious and slowly increasing the doses as data came in about safety and stuff. And then there was a very memorable occasion where you and I, Frank, were both there. And this was at Ionis headquarters in California. You and Anne invited us over to share the first results from the trial. And I'll let you describe what those results showed. But what I will tell you is that it's the first time that a graph ever made me cry.
Yeah. So it was, it was very emotional. Is that we put a lot of we being a very global we, it's not only the Ionis employees, but our colleagues at Roche and all the clinical investigators that were part of the study, as well as the community.
Yeah.
Put a lot of work into getting us to that point. And, we had generated data that, the primary purpose of the initial study was to, escalate the doses, as Ed said, to demonstrate that the drug was safe. And nobody thought that we'd see any clinical benefit in such a short-term study. But the other important aspect was the biomarker. Did we engage the target? And I think that's what brought tears to your eyes, is that we all very excited to see that we got this very beautiful dose-dependent reduction of mutant Huntington in CSF, and the levels of reduction that we saw were significant. That we predicted that we were getting 70 to 80% reduction in cortical tissue based on the CSF value. So the trial was a resounding success, and that gave our partners, Roche at the time, the confidence to go forward into a phase 3 trial. And we could talk about that in a minute, but that was really critical data to show that we're on the track to developing a therapy for Huntington's disease. We hadn't solved it by any means.
No, but I've described what that moment as a something like a save point in a video game, which is if you're killed in a video game, you don't have to restart. You go back to the last time you saved the game. And I think 93 was our first big save point as a community, because from that point onwards, we knew the cause of HD. 2017, I think, was the second big save point, because whatever else happened next, you can always go back to the now fact that we can meaningfully engage with the cause of HD in the human nervous system. And the big bad guy, mutant Huntington, need not always be there in the way that it always has been in the entire human history of Huntington's disease. And that's really something, but spoiler alert, things did not go to plan. And in 2022, so there was a lot of optimism, and we launched a phase 3 trial with roughly 800 people. Phase 3 is the one where if you hit the target, the clinical target, the FDA will give you a thumbs up and you can prescribe the drug. And that trial ended up being recruited in record time, thanks to phenomenal efforts by the HD community. And even during the COVID pandemic, recruitment hit the target so quickly, and every visit more or less was able to be carried out, even though there were rolling lockdowns across the world. But then in 22, was it 22 or 21? It must have been 21.
Yeah, I think it was 21. That's my...
Yeah, the spring of 21, I got a text message from the team at Roche saying, are you available in the next 5 minutes for a call, a conference call? You should also check our website. And so I found out more or less the same way as everyone else, that by seeing the press release on the website saying that the trial had to be stopped early because there were signs of concern, and the safety committee had concluded that there was no realistic prospect that the drug was going to succeed. So that was obviously a huge and terribly sad moment for everyone, made 10 times worse by the fact that we were, broadly speaking, unable to meet in person to figure out what had gone on and commiserate. What do you remember about that time? What were your thoughts and feelings?
Oh, it was... we had so much hope on the drug, and getting that news was, like losing a loved one. It was, very devastating. And the data is the data, so it was hard to refute, or I think the community as a large kind of looked through the data and realized that that was the right decision for the patients that were currently in the study. I'm still confused by, some of the data. I thought we were on target and that we should have seen some, more of a benefit in patients. And it's easy to play Monday morning quarterback and say, this was wrong, this is wrong, this is wrong. And I think we all recognize that there were issues that were occurring that may explain the data, but I still feel that we don't have a really good explanation. And I mean, you're very intimate in sort of the data analysis, and you probably have your own conclusions, but do you have any more clarity about what was driving that?
I mean, I think so. I think I had a working hypothesis as the data emerged, and if you look way back to the results of the first trial, the level of a protein called neurofilament was starting to rise in the spinal fluid, even after those 4 doses in the first trial. And we also had seen some data that there was increases in protein and white blood cells in the CSF, which is generally speaking a hallmark of inflammation or the immune system becoming more active than it normally would be. So it's a bit of a cliche, but sometimes cliches are helpful, that the only failed trial is a trial that we don't learn everything from that we possibly can. And although this was clearly a resoundingly negative trial, I think Roche and the whole team involved in analyzing and interpreting the results have been really actually very open and rigorous in trying to get the most out of the data to figure out what went wrong and why, and then make a kind of hard-nosed decision about whether to I don't like to say this out loud, whether to put the drug in the trash, or whether there's any hope that this drug could be given in a different context that would produce benefit. And I think the big 800 patients is a lot, and a lot of doses were given and a lot of data was generated. So and you can look back and divide that big population into smaller populations to ask what I think is a perfectly reasonable question. Was there a population in which maybe the brain was a bit more resilient? So, we talk about therapeutic windows with drugs. If you go below the therapeutic window, you won't engage with the target. And then inside the window, you get overall benefit. But then if you go over the window, you get more harm than good. So one obvious question was, well, did we miss the therapeutic window? And, you know much more about this than me, but my understanding is that ASOs in general can have a tendency to trigger inflammation in the nervous system for reasons that are not fully understood. We also know that the immune system maybe behaves a little bit strangely in HD, maybe a bit overactive. And the other thing that's obvious is that the brains of people with clear symptoms of HD might be more fragile. So that the hypothesis that immediately sort of sprung to my mind was, well, if high doses of this ASO cause inflammation, and that might be a bit worse in HD, and if the brain of HD patients is a bit more sensitive to that sort of pattern of immune activation, you could see how that could start to cause more harm than good and push you above the therapeutic window. In other words, too much drug in fragile brains. And when Roche did that, what's called a post hoc analysis, dividing the population into basically younger people and older people with more advanced disease, it was fairly clear cut to my mind that younger people with earlier on in the disease were more resilient to this, what in retrospect I think we would be calling an overexposure to the drug. So too much drug in too short a period.
Yeah, and so that's being tested now.
Right.
They're doing a phase 2 study. In the next few minutes, maybe it's worth just chatting about other drugs that are in the pipeline for HD. And you're probably involved at one level or another in many of the clinical studies, or have talked to companies that have designed clinical studies. So there's additional gene silencing approaches that are being taken on, as well as drugs that are in the pipeline that are being developed for masking or dealing with symptoms that the patients may experience. And do you want to just spend a few minutes about the pipeline. And then the real question I had for you is that being involved in multiple clinical trials, oftentimes you have to choose, which patient to match to which drug. And how do you go through that process of helping to navigate patients to specific drugs that you're testing in really essentially clinical experiments?
Yeah, so the pipeline thing first, what's been amazing to me is that the disappointing news that we had a couple of years ago could have been the end of Huntington lowering altogether, but it definitely hasn't been. Huntington lowering is not dead. There are multiple ways of figuring out how to do it, not just with ASOs, of which there are actually multiple different candidates being tested now and developed for future use, but also different mechanisms, including gene therapy involving brain surgery to essentially reprogram the brain to produce less of the protein permanently if all goes to plan. But now we're even testing a pill, which changes the way that the brain handles the RNA in a way that seems to get into the brain and produce Huntington lowering without the need for giving the drug by injection. Which of these is going to work? I don't know, but it's amazing that we've come this far, not just in terms of discovering stuff about Huntington lowering with ASOs, but across all of these other modalities. But wait, there's more, as they say on the infomercials. In the past 10 years or so, studies in thousands, tens of thousands of HD families from Enroll-HD and other studies have revealed incredibly promising new targets that are not the Huntington gene. And these are to do with the way that our cells repair DNA, which we now know plays an important role in the rate at which HD becomes apparent and progresses. So we have multiple targets and also now multiple technologies for engaging with those targets. How we choose people for trials is, in some ways, it's very kind of black and white in that every trial has inclusion and exclusion criteria. And most of our patients who want to be in clinical trials are already in the Enroll-HD study. And each site will be able to consult the data from that study. And that will have information about things like CAG repeat length, medications, severity of symptoms, family history, other, all of the stuff we need to check against the inclusion and exclusion criteria. So then we come up with a shortlist, and we might have 10 people per place in the trial. And so then we have to kind of step back and think about the bigger picture, which is really about things like a trial is really hard work. Everyone likes the idea of being in a trial, but the reality is that you might experience discomfort, side effects, inconvenience. You might have to take weeks off work, depending on the nature of the trial. You might have to travel for hundreds of miles, and you'd be spending an awful lot of time in hospitals talking to people like me, which many people would view as a grave disadvantage. So, the first check is, can this person that we know well withstand the pressure of being in this trial? And then it comes down to practicalities like, if this person has ever had a lumbar puncture, how did that go, if that's necessary for the trial? If this person's ever had an MRI scan, how did that go? And then, a lot of the time we put people in the screening process, they sign the consent form, we're getting all geared up, and then something strange will pop up, like there'll be a blood test result that's not quite right. And it turns out on further investigation that this patient has a rare variation in blood clotting, which means that they can't have lumbar punctures, or that their ECG recording has a measurement that's a little bit too long, and that, that can prevent them being in trial. So there's a lot of, trial and error, to use an obvious pun. The main determinant, though, is enthusiasm and determination on the part of the patient and their family. I won't say that most people who want to be in a trial will get to be in one, because that's unfortunately not true. But the people that want it the most tend to be around long enough and be considered for enough trials that we often find a slot for them. Unfortunately, nothing we've tested in people has worked yet. And so we are reminded that the purpose of all of this is, from the patient's point of view, the purpose of being in a trial is to try to create a better future for other people. And if they themselves benefit along the way, then that's kind of a bonus. That's what we want for everyone, but it's not something that we can promise.
Yeah, thanks, Frank. Thanks, Ed. I think that's a perfect transition to our Q&A session with our participants. Thank you both so much for sharing that perspective. It's a really interesting look into kind of, see how the cookies are baked and where that's really going to get us at the end of the day. So I think I will start with a question for Frank from Jonathan Munkemeyer. And he says, with 6 neurological diseases being treated with FDA-approved ASOs, what scientific issues are delaying ASO progression for Huntington's disease?
It's a very good question, and I mean, part of it is it just takes time to find drugs that are safe and effective, and so Ed mentioned about the DNA repair mechanism. There's a lot of enthusiasm around that pathway for finding drugs, but to find safe and effective drugs, it unfortunately takes years of hard work to get there. So it's those kinds of questions, and that are issues that are slowing it down. It's not for lack of trying. So that the HDF sponsors a number of postdoctoral studies that are helping to advance the science. That advancement in science generates new ideas for how to develop drugs. And then, the pharmaceutical or biotech companies take that on and develop those drugs. And, unfortunately, it just takes time. And I realize that Huntington's disease is a disease where time is a very precious commodity. And so, we're working as hard as we can, but it's not really, you can't point to a single issue that's stopping this from advancing.
One thing to add on that, though, is that we don't get to rewrite history, but we certainly do get to learn the lessons from history. And, I think now, if we were where we were in 2015, 16, 17, trying to design the next trial of a new drug, we'd be paying a lot more attention to things like the NFL, the inflammation, the white cells and protein, and thinking, maybe that's a sign that we should approach this more cautiously or more patiently, perhaps. And again, I think that that comes back to the only failed trial being one that we don't learn from, and that we need to put those lessons into practice.
Yeah, absolutely.
That's a great point. So this is a question I'll direct toward Ed. An anonymous attendee asked, you mentioned that clinicians were not initially receptive to intrathecal dosing. How has that changed since the tominersen trial started? And how receptive are patients today at the idea of regular intrathecal dosing?
So I would say that most of the skepticism about that particular aspect of this program is gone now. One of the great pieces of data that we got from the tominersen program was that thousands of intrathecal injections were given, and there was no suggestion that the actual process of injecting stuff into the spine and the spinal fluid of an HD patient was harmful. If you look at the placebo group, treated patients in that program, they essentially progressed exactly as you would expect according to natural history. Their NFL didn't go up. There were no sort of terrifying sounding adverse events related to injections. That we have to, when we consent people, we have to talk about the minuscule risk of things like bleeding or nerve damage or infection being introduced into the CNS. And that basically didn't happen in the Generation HD1 trial. So I think now the community's just and we've also seen other drugs that Frank and his colleagues have invented being licensed and really just changing the world for kids with SMA and people with various forms of motor neuron disease and others. And so I think that that principle is now kind of very well established. It's making its way into clinical practice in those diseases throughout the world. And as time goes on, I think we will start to see this in spinal injections as quite routine. And they're certainly now in the HD Centre here in London, a perfectly routine part of the training of any new doctor who joins the team.
So Ed, you actually volunteered to essentially do a spinal injection. We didn't inject anything, but you, I thought, donated spinal fluid for a study. And
yeah,
was it scary for you?
I did that a few times, and it was back in the day when even collecting spinal fluid was not routine at all, and it was something that a lot of people were understandably terrified about. So I basically having decided to collect some samples of CSF for the biomarker program, I decided to do a bit of kind of education. I went on Twitter and I live tweeted my lumbar puncture. I'm not on Twitter anymore for hopefully obvious reasons, but anyway, it's I don't know if the tweets are even still there, but I tweeted it, and then I think I had 3 more lumbar punctures, partly just because it was quite difficult to get CSF from control volunteers, but also because it genuinely wasn't so bad. I'm not good with pain. I hate having my teeth even cleaned by the dentist. I do it myself at home. I'm very brave about that. But anyway, having the spinal fluid collected was, in my personal experience, not too bad at all. And we like to create quite a relaxed atmosphere for the patients. We play music, we take things nice and slowly and relaxed, and we're kind of in a groove now.
Awesome. Thanks, Ed. So I'll send this one to Frank. This is from Melanie Wellmill. And so I think this really speaks to what you guys were both saying about learning from other trials. And obviously, in a world of COVID, we saw vaccines being developed in what felt like a matter of months. And so she asks if the speed of the COVID vaccine helped fast track any of the HD drugs. And maybe you could talk, Frank, about if the development of the COVID vaccine changed how you saw drug development proceeding.
Yeah, so maybe it's worth just sort of putting that rumor to rest, is that the COVID vaccine actually was building on a big story that was going on with RNA-based vaccines that was over a decade in the making. And so there was a lot of work by Moderna and other companies, BioNTech, that were developing RNA-based medicines for vaccines for cancer or vaccines for viral infections that had happened over a decade. And a lot of the testing in patients had already transpired. So they knew that the technology was, safe and well tolerated. And so when COVID happened to come on us, both those companies sort of dropped what they were doing and recognizing the impact to the world, focused their whole organization on developing vaccines for COVID. So it was really building on a very strong foundation to get us to where we were back in 2000. So just to clarify, it wasn't brand new. It was a lot of knowledge that led up to that, and then it was sort of tweaking the formula, the recipe at the end, adding a little bit more sugar or something to make a better cake. But it was well done. So I think as a society, we have to recognize how everybody pulled together to find a therapy for COVID. And to me, that was as a human being on Earth, that was the part that I really was very proud of as a person in our society, is how the world pulled together to solve this problem. And it gives me confidence that for a disease like Huntington, that's possible. It's not going to be next year. It's not going to be in perhaps 5 years, but it does give you confidence that if people really pull together and focus on solving a problem, they're very capable of doing it. Ed, you wanted to comment as well on that.
No, I completely agree. I was going to say the same thing. It was about people's mindset changing because they suddenly realized the intensity of the threat. And it's kind of frustrating because I think that the HD community is small, but those of us who have the privilege to operate within it have the same feeling of the intensity of the threat and the time critical nature of the need to do the work we are doing. So we're limited, I think, by resources. And the other big story about the COVID vaccines was that governments and companies alike put tons of resources into dropping everything else and putting the foot on the gas. The third piece of that jigsaw, I think, was new ways to collaborate, and the idea of you would develop a drug, you'd patent it, you'd wait for the paper to come out in the New England Journal of Medicine, and then on the basis of that, the community would start to be interested in running trials or whatever. Not quite like that, but you get the idea. Suddenly everyone was sharing all of their initial data online, and new sequences of the virus were being published, and governments were rolling out new platforms for enrolling people into tens of thousands of people clinical trials. It just, I don't know. I do share your really strong positive feelings about that, but in a way it makes me slightly sad that we don't do all science like that. We should be spending 100 times as much money on science as we are, and we should all be collaborating in the ways that we did during COVID. A lot of it has stuck, so there's a lot more publication of early results and so on. But we learned some,
and I have to
pieces, I think.
Have to say, I've been in a variety, I've been at this for a long time and worked with a variety of different disease areas. And I have to say the Huntington community is really one of the most collaborative communities I've ever worked in. You have a large number of people, scientists that are all pulling in the same direction and readily share data, readily share ideas. Sometimes we're hypercritical of each other's experiments or designs, but that's a normal scientific process. I think everybody wants to see, therapies be developed for Huntington's disease.
Thanks, Frank. So I think I'll send this one to Ed. This is from Natasha Beal. She's curious about the PTC 518 trial. She wants you guys to talk about the development, and she said she thinks that there was data collection that ended at the end of April 2024, and was wondering if there's going to be an update soon. Do you know about that?
I mean, the one thing you can be sure about in regard to any trial you haven't heard from recently is that there will be an update soon. Different companies have slightly different, I guess, cultures as regards giving regular updates and what they will and won't cover. PTC 518, which is the pill that lowers Huntington, which I mentioned, we found out last year that it does lower Huntington in humans, human patients with HD. What we are waiting to hear now is updates from more numbers of people and somewhat longer term treatment. But again, this is a first in human trial, so it's quite small, and even the final results of that trial will be somewhat provisional and subject to further prolonged investigation. I think we are expecting an update soon. What I would say is that if there's a data cut, if the sort of last patient, last dose, or last patient, last visit is in April, it will take several months after that to clean the data. That's not like faking the data. It's basically the independent monitors for the trial coming in and saying, you missed a measurement here. Can you go back to your records and make sure it's there? Or this number was different on the electronic record from what you wrote in the paper record, and all of that sort of going through the due diligence and making sure that the data that end up being analyzed are completely definitive and as complete as possible. Then they have to send all the samples to the lab. They run the samples in batches for most of these tests, like Huntington and neurofilament. So they don't run them as they go along so much as they have to wait until all the samples are in the freezer in one place. Then they run them in a batch, and then all of that has to go through rigorous quality control. All of this goes to say that the last patient, last visit is going to be probably at least a couple of months before the sponsor is in any position to make a public announcement, because the last thing you want is to make an announcement that sounds great and then they have to retract it, or sounds terrible and then it turns out, the share price tanks, and then it turns out that was all a big mistake. So soon, yes, but what the meaning of soon is varies between one setting and another. But I believe that we can certainly expect an update within the coming few months, let's say.
Thanks, Ed. So I think we probably have time for one more question, and I think it would be nice to hear from both of you guys on this. From an anonymous attendee, they're wondering if you think the treatment for HD will be a cocktail of medications.
Yeah, so I do. I think given the disease and what we know about the disease, it's unlikely that one drug is going to give us the benefit that we would really like to see in the patient. So I think it may take a couple of drugs to do it. In addition, some of the drugs that are being used to treat some of the symptoms would be part of that cocktail. But I do view that it's likely that we're going to be thinking, like in cancer, that it's really being treated by a cocktail of drugs that are really tailored for the individual patient.
I agree. I think that's almost certainly where we're headed. We need to before we can have a cocktail, we need one drug that works. We know that tominersen does work at this at the kind of molecular numeric level. And what that reminds me of is where we were with AZT in the early 90s with HIV. AZT was the first antiretroviral drug for HIV, and there was great excitement because short-term treatment with AZT was shown to reduce the viral load, the amount of virus that was in the blood. But unfortunately, prolonged treatment with AZT started to cause damage to the liver, and then the virus started to become resistant to it. And so it's a really valuable example of how changing the numbers can represent progress, but doesn't guarantee success. And what really brought success with HIV was the advent of combination therapy, multiple drugs coming at the same problem from different angles, but also tailored to the individual. What strain of HIV do you have? How did you respond to this? This drug messed up your liver a bit, so we put you on a different one. When we have multiple drugs that work a little bit for Huntington's disease, I can imagine us using, for example, a gene therapy in early life to really reduce Huntington production in the striatum, the very central structures of the brain that are affected early on. Then an ASO injected into the spine as a kind of intermittent, maybe top up, and we might start to talk about Huntington control, just as we talk about glucose control in diabetes. And then why not a pill as well? So you take a pill every day, you've got your gene therapy chugging away in the background, and all of these DNA repair drugs, maybe those are stuff that will take priority very early on in life to try and prevent the onset of the disease or the progression that we see at that level in the brain. So great question. The answer is yes. All we need is several drugs that work.
Thanks, Ed. All right, well, we are at the end of our time, so I'm going to wrap it up there. If you had a question that we didn't get to, and we did have quite a few questions that we weren't able to get to, I'll send those to our speakers so that they can follow up with you directly if they're willing, and I can also reach out to some of you on the easier ones that I won't bother Ed and Frank with. So I want to end by again thanking our sponsors for this webinar, Neurocrine Biosciences, Cleveland Clinic Lou Ruvo Center for Brain Health, and Unicure, as well as everyone who donates to the HDF to support the research that we're able to fund, our audience, and last but certainly not least, our speakers. Thank you, Frank and Ed, so much for being here. Our next webinar is Thursday, June 13th. We'll be joined by Dr. Juan Botas from Baylor College of Medicine. He will be sharing his work that looks at genes from people that live past 100 years old to try and better understand Huntington's. So thank you so much for joining us today, and we hope to see you next time. Thanks, Ed. Love the fireworks.
Bye.